Okay. Hi, everyone. If you could go ahead and sign in for me. This is just so people know that I'm doing my job and that you're watching my study sessions, like if you can't make it in person. Um, this review is going to be really quick. It's a lot of terminology, and I have really bad handwriting, so I just left it as print. So we're gonna get through the material pretty quickly, but go ahead and like pause it and like write down and take notes or like screenshot it just so you can read it after. Um, with that being said, okay, let me make sure this works because last time it froze. Okay, phosphodiester linkage is the linkage that forms the backbone of DNA. Okay, ester bonds are phosphate groups that link two sugar molecules by, well, they link two sugar molecules. A phosphate group links two sugar molecules, and a nucleotide is a base, sugar, and a phosphate, and then a nucleoside is just a sugar and a base. Okay, purines, I'm gonna use laser. So purines are two-ringed bases, and it's our A and our G. Pyrimidines are our one-ringed bases, and this is T, C, and U. Okay. Now, what is Chargaff's rule? So this basically is just saying like the A's are equal to the amount of T's and the C's are equal to the amount of G's. And you can see this multiple ways. You can see it like this too. As long as like the base pair is on the other side of the equal sign, it's right. Just be careful. Remember your base pairing A to T and C to G. Um, one strand of DNA is what is the opposing side, what is the opposing strand? How many hydrogen bonds are present between the two strands? Okay. So what I always like to do is we know DNA is anti-parallel, and I'm going to draw straight, like, through the question. But so this is going to be my 3 prime, and then this is going to be my 5 prime, because we know DNA is anti-parallel. So then we're just going to do the base pairing. So that is going to be the opposing strand. Just remember, they might be flipped. So make sure your 3 prime and your 5 prime line up with your answer. So how many hydrogen bonds are present between these two strands? So we know our AT bonds have 2 hydrogen bonds, and then our C's and our G's have 3 hydrogen bonds. So you're just gonna count 3, 3, 3, 2, 2, 3, 3, 2, 2, 3. 3, 6, 9, 12, 15, 18, 19, 20, 21, 22, 23, 24, 25, 26. I believe that's 26. Don't quote me on that. I would do the math. Um, one strand of RNA is the following what is the opposite strand. So I'm basically going to do the same thing. Here is our 3 prime, and then here is our 5 prime. Oops. He even got me. U G A. Okay, so again, this question is just trying to get you to remember your U's here and here, and also remember your primes. Okay, what are key differences between DNA and RNA? Structure, DNA is double-stranded. The sugar is deoxyribose, and that is because carbon 2 does not have hydroxyl. Our bases for DNA are A, T, C, and G. And then our location is in the nucleus and mitochondria. For RNA, our structure is mainly single-stranded. Our sugar is just ribose, hence the R in RNA. Our bases are A, U, C, and G. And our location is in the cytoplasm. Okay. And then I didn't make this review. This review was already made. So I don't really know what the person meant by 2 strands of DNA forming a double helix can be described as. They either meant they could, yeah, they could have meant a lot of things, but I took it as anti-parallel or complementary. But really, there's a lot of things that could finish that sentence. Um, anyways, DNA exists in a double helix. One characteristic of the structure includes parallel strands, which we know that's wrong because DNA is anti-parallel. Counterclockwise, we know that's wrong because DNA is clockwise. 10 bases per twist. That sounds right. Let's just see what our options are. Purines based with other purines, we know that's wrong because purines base with pyrimidines. I would spell it wrong. And then last option is 4.9 nanometers per complete twist. That's wrong. The answer is 3.4 nanometers. So we know our answer is going to be 10 bases per twist. Okay, chapter 12. Euchromatin is transcriptionally active and less condensed. One trick I learned is like the U in euchromatin means that you can use it. It's transcriptionally active. Heterochromatin is just the opposite. It's um unactive and more condensed. So chromosome territory is a region in the cell nucleus that occupies a single chromosome. Telomeres are DNA sequences found at the ends of linear eukaryotic chromosomes. Remember, they're so we don't, when we make more DNA that the ends don't fall off of our chromosomes, we need all of our chromosomes, so we kind of put um, sequences at the end so none of it falls off when we make more. Centromeres, we know this. A region of the eukaryotic chromosome that provides attachment for the kinetochore. Remember metaphase, centromeres? Anyways, loop domains, a segment of chromatin that is organized into a loop by CTCF proteins. Nucleosome, and this is mainly asking about composition. It is composed of double-stranded DNA wrapped around an octamer of histone proteins. So this is like our beads on a string. Transposon is a transposable element in which DNA is removed from its original site and transferred to a new target site. Retrotransposition is a form of transposition in which a transposable element is transcribed into RNA, and then RNA is used as a template with reverse transcriptase to synthesize a DNA molecule that is integrated into a new region of the genome via integrase. Okay. LTR retrotransposons, a type of retrotransposon that is evolutionarily related to the retrovirus and has long terminal repeats, hence the LTR. Insertion elements, a transposon that moves via simple transposition and carries additional genes like inverted repeats and the transposase gene. Okay. 7 copies of a retrotransposon are created and integrated into the genome. What is the result or outcome of this retrotransposition event? Okay, so like I said before, retrotrans transposition is where I like to think of it as copy and paste. So here's our, yeah, I'm just like, here's our DNA, right? And then on this segment, this is our gene of interest. I'll just leave it like that. That little part is our gene of interest. So what happens in retrotransposition is that we copy it. So we take this, turn it into RNA, and then the RNA is used as a template with reverse transcriptase. So just imagine reverse transcriptase is doing its job, synthesizing a DNA molecule that is integrated into a new region of the genome. So then this DNA, which is exactly the same as this one, these two DNA are exactly the same. It just took some extra steps to get there. And then now we're going to take this DNA and we're going to put it here. And then we already have a copy of DNA. We're just going to put it here, further down the line. So in the question, 7 copies were made. So the total would be 8. So 8 copies. Because you still have your original. You never took this piece of DNA out. It still stayed there. You just copied it into RNA and then DNA and then put it different places. So like just remember copy and paste. So what are the levels of DNA compaction? Least compacted to most compacted. So obviously we're going to start with naked DNA and then we move to nucleosome. Then after that we go 30 nanometer fiber and then loop domains. And then we have a fully condensed chromosome. Okay, chapter 13. Topoisomerase 1 relaxes negative supercoiling, and topoisomerase 2 relaxes positive supercoiling. Single-stranded binding proteins keep the parental DNA strands from reforming the double helix. So, I mean, the name implies what they do. They bind to a single nucleotide to keep it from binding to the other side, to keep it from snapping back together. Primase synthesizes RNA primers for the lagging strand. DNA polymerase 1 is an enzyme that fills in regions of DNA where the RNA primers were. So it's kind of the cleanup crew. DNA polymerase 2 plays a role in DNA repair. Um, DNA pol 3 synthesizes DNA in the leading and lagging strand. This is the star of the show. This is the one we talk about the most. But flap endonucleases, so that removes RNA flaps from RNA primers on the Ozaki fragments. And the Ozaki fragments are just the short DNA that is synthesized from the lagging strand. So remember, we have to go from 5 to 3, and we have to do that in bits with the lagging strand. So these are those fragments. Um, ARS elements are DNA sequences found in yeast that function as the origins of replication. Telomerase is replicating the 3' end of the chromosome. So these are our enzymes that keep our telomeres, like they upkeep our telomeres to make sure they're at the right size. Um, Rec-A proteins promote strand invasion and D-loop formation in the double-stranded break model. Okay, now what enzymes or biological molecules make up a primosome? So what makes up a primosome are the 6 proteins, and they track. And synthesize RNA primers. Okay. DNA is synthesized in 5 to 3'. while DNA is proofread in the opposite way, so it's proofread in the 3 to 5 prime. Okay. DNA helicase enzymes move in what direction? So helicase moves in the 5 prime to prime direction. What is the difference between DNA polymerases used in prokaryotes and in eukaryotes for the replication? So DNA pol 1 removes RNA primers and fills in the gaps. And DNA pol 2, DNA primarily does DNA repair. DNA pol 3, this is our main guy who synthesizes. Sorry, guys. Um, synthesizes synthesizes DNA in leading and lagging strands. Okay. And then we'll go into alpha, beta, and gamma. Okay, and then alpha initiates DNA replication. Beta plays a role in DNA repair. And gamma helps replicate mitochondrial DNA. Sorry, there's a dog outside. I don't have a dog, I swear. Now for chapter 14, introns are a segment of RNA that is removed during RNA splicing. Exons are a segment contained within RNA after splicing has occurred. In mRNA, the coding sequence of a polypeptide is contained within the exons. Alternative splicing, this is really important, allows several proteins from a single gene. Tailing is a string of adenine nucleotides at the 3' end of eukaryotic mRNAs. And this is really for stability of the mRNA. 5' capping, important for the initiation of translation. FTIIH act as helicases and promote the formation of an open complex. Sigma factor belongs to a general group of proteins called transcription factors. I would look that one up again. That's kind of a broad definition, but I think that's from the book. So, one DNA strand is following what is the complementary RNA strand? So what I like to do is the same thing, but I'm actually gonna do it the other way because you're most likely gonna have the 5 prime on this end because it's asking for the complementary strand. So it's gonna be U, U, C, G, A, A. So just make sure you check your values like this. Come on. Like that is there, but you have to remember our primes. Okay, what are the 3 steps in transcription? So this one's kind of a gimme. Initiation, elongation, and termination. Okay, understand how splicing works with introns and exons. If splicing sites on the intron were not recognized or repressed, what is going to happen? So if an intron is not removed and is still in the mRNA, this can lead to a frameshift mutation. And then it can just mess up the whole thing. So like you can have stop codons where you're not supposed to. Immature stop codons. Or you can like produce non-functional. So one intron can really mess up the whole protein and like all the proteins. Okay. Protein is a functional unit composed of one or more polypeptides. tRNA is a type of RNA used in translation that carries an amino acid. It's important. Carries an amino acid. Each anticodon is in a tRNA and a complementary codon in the mRNA. A charged tRNA has an amino acid covalently bonded at its 3' end. And the anticodon that we were talking about earlier is a 3 nucleotide sequence in the tRNA that is complementary to a codon in the mRNA. Ribosomes is composed of structures called large and small subunits. And the nucleolus is a region of the nucleus. 30 and 60 subunits are assembled there and are exported to the cytosol and associate there. Now we were talking about this today, the Shine Delgado sequence. It's a sequence in bacterial mRNAs that function as ribosomal binding sites. Now, the peptidyl transferase complex is a complex that functions during translation to catalyze the formation of peptide bonds between the amino acid and the A site and the ribosome and the growing polypeptide. Now the wobble base is the third base. The third base is a degenerate base. For example, these all code for the same amino acid, but they all have a different third base. So the third base can kind of be pointless, because you end up with the same thing. That's why it's called the wobble base. Like it could go like either way. Um, a C terminus composition, the last amino acid, it has an exposed carboxyl group. Okay. What are the three sites of a ribosome? So this is our P, A, and E. Our P site holds the tRNA. Our A site um brings the next amino acid to like elongate the to add to the growing chain. Um and then we have the E, which is the exit site. This is uncharged tRNA exits. Okay. Now Kozak's rule. Um, and these are just favorable, um, fa- so we usually have a guanine at the +4, and then we have a purine, which can be A or G, um, at the -3 position. Okay, the ribosome is composed of multiple subunits belonging to the 30S ribosomal subunit. What is the role of 16S rRNA? Um, so this is, it has the, has a complementary sequence to the Shine-Delgado. So this helps with the initial binding. Okay, what does RNA polymerase 1 produce in eukaryotes? And it produces 18S rRNA. Okay, last but not least, know your amino acid chart because you will use it later. Like you use this in biochem again. Um, so just look at your labels, first base, second base, last base. So let's say we're doing UCA. So we're gonna start off at U because U is our first base. Then we're gonna look at our C because that's our second base. Then we're gonna go across here and look at our A. And what do you know? They all code for serine anyways. So just know how to use this. Practice it a couple of times. I know it seems like common sense, but you'd rather practice it and get it right than lose points on an exam. Um, but anyways, if you have any questions, just let me know.